taking the time to see what we've been up to. Let me just share the screen. Can you all see that, Hartwell and Joseph?
Yeah.
Okay.
Yep.
Cool. So yeah, thanks for being here. We've done some cool work, and Joseph's going to help us explain it. But let's get going. A couple of logistical things. We're going to record this webinar so that we can share it for folks who can't be here right now, that were interested in being here. So that's one thing. And then as folks have questions, feel free to put them in the Q&A box that you have at the bottom of your screen. And we'll have a short session after the presentation to kind of tackle those. Cool. So we've been doing some what's called source tracking in the French Broad, which is just DNA analysis of the water samples that we've been collecting. We're MountainTrue, so we're an environmental advocacy organization based in Western North Carolina. We focus on a lot of different issues, including watershed-based stuff, and land issues, energy issues, as well as land use planning. Jonah Ventures is the lab we use to help analyze our water quality samples. And Joseph is here to help kind of explain that, and feel free to throw in more information about your lab when we get to that, Joseph. Sorry, my computer's slow to move. Well, let's try this. Anyway, I think the next slide is introducing the two of you anyway. So Hartwell Carson is here. He's our French Broad Riverkeeper. Oh, there it is. Cool. He's our French Broad Riverkeeper. He's been it for forever. He knows everything about the watershed and is an expert in water quality for the French Broad. And then Dr. Joseph Craine is our scientist here to help us explain the process and the lab piece of what we were doing with source tracking. So I'll let Joseph take it from here.
Thanks, Anna. Can you hear me? You can hear me. So I can hear me, you hear me. If you want to mute your mic, Anna, just to make sure there's no feedback. So, so thanks to Hartwell, thanks to MountainTrue for having me here today and everybody in attendance. What I'll talk about a little bit today is some of the work that we're doing in water to help people understand sources of fecal contamination. And that approach that we're using in general is called source tracking, because you're tracking the source. It uses a molecular technique. And leaving out some details, essentially what we do is that if we detect bacteria in water that's likely coming of fecal contamination, then we want to know what host that material is in initially. There's a couple ways to do it, but what we focus on is that we're going to take a look and try and quantify the amount of DNA that is coming from the mitochondria of the host. So it's going to be host specific, and it gets passed out in fecal material. So you look at human poop, there's human DNA in there. You look at pig poop, there's pig DNA, etc. And then we just take a look at how much of that is in the water. The technique that we use is called quantitative PCR or qPCR. That sometimes you might have heard this recently in the news, the qPCR technique, because in the COVID world, when they're doing these tests, the gold standard test that they're doing now to detect whether somebody has COVID or not are qPCR techniques. And essentially it's a quantitative technique to quantify how many copies of DNA are present from a particular target. Next slide, please, Anna.
Sorry, it's taking a minute.
All right. So we put together a little bit of a graphic on the two different approaches that people use for source tracking, just for some background. And one of those is to take a look at the bacteria in water. We know that some of that is going to be E. coli, but then there's a whole bunch of other bacteria that are in the water. And one approach for source tracking is bacterial source tracking, where we then quantify using the qPCR technique how much bacteria of a certain species is present. And this particular bacteria would be an indicator for a host. So there's a chicken bacteria and a cattle bacteria and a dog bacteria, something that is more likely to be associated with one particular host. Then we go into the water, take a water sample and see how many copies of that particular indicator bacteria are present. That technique works just fine to a certain degree, except when it doesn't. And one of the reasons that it might not work is that not all hosts, not all cattle, not all dogs have the same microbiome, the same microflora in their intestines. And actually, a large proportion of the time, that indicator that people use for a specific species can't be found in that host. So you go to one herd of cattle and they might have it, another herd might not, and therefore you just would not find the indicator. Our approach for looking at the mitochondria of the host is that it's going to pass, it's present in every animal of a given species, and it passes through into the fecal material. So it's a lot more diagnostic for determining the source of the material. So if we have cattle and their fecal material gets into the water, then they're also going to pass some of the mitochondria that come off of their large intestines, small intestines, things like that, gets passed at the same time. So that's the technique we use. It's mitochondrial based, it's source tracking, but it's a little bit more specific than some other older techniques. Okay, Anna. Great. So in this slide, it's just, we've done this a bunch of times and we're showing that we're going to get different results depending on what the sources are. So when we went to Alabama and did this work, that we can look at our different samples. The data that we get back is we start counting how many mitochondrial sequences we find from the different hosts. So I don't know how we can't see, you wouldn't be able to see my cursor. So for the, in the upper right-hand corner, it says bovine, which is cattle, dog, human, poultry, and swine. That first sample was relatively low across the board. Second sample, we had a little bit more dogs, so there would have been a little bit of dog input on that one. Third one, pretty much clean. None of the potential hosts really showed up. And then the fourth one was sky high as far as human is concerned, but then you also find a little bit of cow, a little bit of dog, poultry, and swine. So that fourth one was a trailer park that had a septic system that was likely leaking. The reason you find human is that there's a lot of human fecal material that's getting into the water, and with that, a lot of the DNA from the people. The reason you find dog is there's dogs there, but pigs, poultry, and bovine, smaller amounts of that. The reason that that's there is not because people are keeping cattle, chickens, and pigs, but it's also an indicator of what they ate. So when that material, when we eat food, DNA from our whatever we ate, our diet actually passes through the system too. So we're going to get lesser amounts of the food that people ate, and it also enters in through garbage disposals and things like that. But if you had a big barbecue and it was everybody was eating ribs, there's going to be a lot more pig in there than you would if it was a big barbecue and everybody was having chicken. I think the panel on the left were some of the ones from the current study. I'll skip over that. But broadly, you can see that there's a lot of cattle and not as much of other stuff, except for one sample that had a lot of human. So it's just qualitatively different pattern. And then we can get to another site that is next to a place that people have a lot of dogs running around. And for the most part, that was the main source of DNA into that system. So we do, when we get into different places, see a lot of different patterns. And what we're showing, we'll be showing here more specifically, is not something that we get all the time. Okay, Anna. We've also tried this in other places. So for example, not only can you go to a single point and see what the results are getting, is that you can work your way down a stream in order to identify where a source enters into the water. So our labs in Boulder, running through Boulder is Boulder Creek, and sites 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 take you from essentially the foothills around Boulder through the city itself and then down out to more agricultural areas. Points 1 and 2 were basically up in the foothills, really clear. That's Boulder Creek where it's nice. Point 3 is when you get a little bit downstream of campus and human DNA just skyrockets. And so somewhere more than likely in there is a source of human DNA that is probably an old septic line in the campus that's leaking into Boulder Creek. And there's some other explanations, but that's a likely interpretation. And then lesser and lesser amounts as those inputs dissipate. We get to site 10, and by then we're out into the country for all intents and purposes, and you've got a lot of cattle actually grazing even within Boulder County. So another approach, not just looking at sites, is we can start to take a look across along a river or a stream. And as we do that, where things start to spike are ways that you can start to see when specific inputs are hitting the water. All right, Anna. Looking at these levels, so if we're going to start to say, okay, is 10 high, is 100 high, is 1,000 high? This is just a scatter plot of what we see across. At one point, we just took all the data that we've gotten across sites. And just to show you that it's not always going to be human. Often there's a lot of cattle DNA. We do get high dog samples, sometimes high chicken, over 1,000. And when we do find pigs, it's often a lot. Whether those are domestic pigs or wild pigs, feral pigs is something else. Sheep, we've looked for that all the time and we barely find anything. I think somebody ate a lamb chop once and that was really it. So when we start to take a look at these levels, 100 is starting to get moderate for copies as far as we can tell empirically. By the time you're getting close to 1,000, you're up at the upper end of what we see. So kind of use that as a scale. 10 is low, 100's in the medium, 1,000 is pretty high. Hey, Anna. I think this is you, Hartwell?
Yeah. So to take a step back, I just kind of want to give you guys a sense of how we got to using source tracking. And particularly in our instance in the French Broad watershed. So for 8+ years, we've been doing E. coli sampling around the watershed. We've been doing it at about 30 something sites, and we've been doing it weekly from like May to September. And we post those samples to a site called the Swim Guide, and it gives the community an idea of, is this stream safe to swim or not at any given moment, and what's the kind of the averages over a period of time. So it's very informative to the public as far as how they can use the water, when they want to use their waterway. But it also really helps folks like me and Anna figure out where are our worst sites and where we should be doing a lot of targeted follow-up monitoring. So we use the Swim Guide for a couple different reasons. The shortcomings of the Swim Guide, though, there are a couple, but one is that, it tells us that we have high E. coli or not, but it doesn't give us a sense of why we have high E. coli. And what we're seeing in the French Broad, and this is a pretty representative sample. These are red is considered bad. And what we're seeing in the water quality in the French Broad is we're having very frequently, most of our sites are failing EPA safe standard for E. coli. And we're seeing that trending in the wrong direction too. And we were getting frustrated at the water quality in the French Broad, and we wanted to do more than we were doing to just monitor it and do our follow-up sampling to look for problems. We wanted to take a broader perspective and then incorporate the scientific analysis of that into some really targeted solutions to improve water quality. So the Swim Guide is a great tool. The source tracking is kind of the next step in that to get us to the point where we can confidently say, here's why the E. coli is this, and here's what we need to do to fix that. Just to give you guys a few examples, Hominy Creek, it has always been one of our worst sites, but it's been trending in the wrong direction. The French Broad through the River District in Asheville is really disappointing. I mean, it used to pass EPA safe standard 60, 70, 80% of the time, and lately it's been failing 60 or 70 or 80% of the time. So we really are moving in the wrong direction. I think there's a few reasons for that, and we'll kind of get into those as we go. You can switch it in. So our old model was take swim guide data, look at the bad sites, and then do targeted follow-up monitoring. And what I mean by that is literally going upstream of, say, Hominy Creek and testing dozens of sites to see if you can bracket where the problem source is. You take 3 or 4 samples downstream and 3 or 4 upstream, and in the middle, in theory, there'd be a point where the E.coli is high and upstream it would be low, and your source is in the middle there. So we did a lot of that, and it is effective, but it's incredibly time intensive because you're taking dozens of samples over a period of weeks, if not months, in a variety of weather conditions. And then you're literally going out and walking streams to try and locate the sources of pollution. So this was one source that we found years ago. This was a dairy farm that was directly discharging their waste into the river. This is kind of an extreme example where they're actually piping waste directly into the river. We were able to fix this, and that's great. But it takes an awful lot of time and type of monitoring. You can switch to the next one, Anna. Another example is this was a straight pipe. This was a busted sewer line, in the River Arch district. And me and Anna probably spent 6 months of sampling, probably hundreds of samples to finally locate the source of this busted sewer pipe. And it sure, looking back, it sure would have been helpful to have taken a sample, downstream to have known we were even looking for sewer. We could have been looking for, some other kind of source. So that's just an indication of the model we've always used, and we still use this because it's effective, but we're moving towards some new tools that we think are going to be even more effective. You can go to the next one. That was me standing in a storm drain and throwing me the sample. So the water quality in the French Broad was so concerning, and it was getting so much attention that we actually received some funding from the General Assembly. Senator Chuck Edwards was really key in getting us some money to do additional follow-up sampling and to use a few other tools, including infrared images from the air that we're using to track sewer and septic leaks, but also for this source tracking piece of the puzzle. And so we targeted 4 or 5 sites kind of intensively, but we also wanted to get a broader picture of the watershed. So we did do kind of watershed-wide surveys. We did dry weather, we did wet weather, and each site varied a fair bit, but kind of the theme of the watershed was fairly consistent and was a little surprising, and from what I thought it might be. You can switch to the next now. All right, so this is kind of the charts that Joseph was just showing, but the 4 sites that we really did more sampling on than any other were Hominy Creek, which is kind of the mouth of Hominy Creek there, which drains Canton and Clyde and West Asheville. We did Pearson Bridge, which is the bridge over to kind of Richmond Hill, just at the bottom of kind of the River Arch district on the French Broad River. We did Cane Creek, which drains Fairview Fletcher area, and we did that at Fletcher Park, so towards the end of Cane Creek. And then we did Mud Creek, which drains most of Hendersonville area. And if you had asked me before we did all this sampling what we would have found, I would have said we would have found mostly cow and human. And that is mostly what we found, but I was surprised that we found more cow than human. And Joseph, feel free to chime in on any of these results too if you want to. But each of these sites was a little different, but cow, really was more predominant than anything else and more consistently predominant. And we do have some spreadsheets of the data too. So if you guys have any specific questions about sites, one of the sites that I thought was even more helpful was Cane Creek, and I'll talk a little bit more about that in a minute. But me and Anna had spent a good bit of time there looking for sewer and septic problems, and as you can see, from that chart on the bottom left, we really should have been spending that time looking for problems with cow, because there isn't a whole lot of human input for the most part from our samples in Cane Creek. You can switch that in. Oh, I also think it's that one site on Hominy things is we had a pretty big hit on dog there, and that was not that surprising because we're just downstream of the dog park there and the Hominy Creek watershed. So I think that's interesting, and that's also after a rain event, so not surprising that you're going to have more runoff during that time period. So it's a handy tool to know that we need to be looking at cow in the Hominy Creek watershed. We need to be looking at human too, but also, you know, after rain events, dog can be a pretty fair contributor there as well. And so looking at management practices, particularly probably in that Hominy Creek Greenway corridor, would be helpful in improving downstream water quality in Hominy Creek. All right, you can go in. And then this is just by county. The graphs are a little misleading. Well, some of them are, because we have just more data in some counties than others. You know, when I looked at this, I was very surprised to see Transylvania County showing so much swine. I thought, wow, that's crazy. We don't have a lot of pigs in the French Broad watershed. You know, Joseph did kind of hit on the fact that you can get swine from what folks are eating, but I looked at the data in Transylvania County. We actually only had 4 samples, and one of them was really high for swine. The others had 0 swine. So that one graph is a little misrepresented on the amount of swine. I think swine is kind of a question mark, and Joseph, feel free to chime in if you want. I'm a little puzzled that we found as much as we found, except for people do really like barbecue in North Carolina, and so that's a fair point. I think this is kind of one of those you know, research questions that I'd like to go look into a little bit further, and hopefully we can do that with some additional funds to do some follow up on some of our sites. But, you know, as you can see here, kind of the pattern that I showed you before, cow is the predominant factor in each of the counties, except for that kind of misleading swine number in Transylvania. And that really is informing our advocacy going forward. You know, we're still definitely working towards and advocating for better sewer practices. And that is key for sure, because you're still getting a fair bit of human, but we also realize we really need to work. We really need to, you know, improve and expand upon our advocacy for agricultural practices too. So I kind of mentioned this Cane Creek problem earlier, but this just kind of gives you kind of a closer example of, you know, Cane Creek to me was one of the real key examples on why this was such a valuable tool. You know, me and Anna were actually taking monthly follow-up E. coli samples from kind of the Fletcher area based around where their sewage infrastructure was. And we were looking for sewer leaks. You know, my theory was it was the town of Fletcher's system was leaking. We were sampling just downstream of the town of Fletcher, and that's why Cane Creek was one of our problem sites based on our swim guide sampling. What this shows, though, is me and Anna should have been spending our time upstream looking for agricultural issues and working with our soil and water conservation districts to try and fence out cattle and provide alternate watering sources and hardening stream crossings so we can prevent the impact of cattle in the Cane Creek watershed. And that's definitely what we'll be doing going forward. While, you know, human is definitely still an impact, and there are still some sewage and septic issues in the watershed, cow is obviously the major problem. You can keep going now. So that's kind of hinting at, you know, where we're getting to MountainTrue wise, using source tracking to really hone what we think the solutions are. And we've become at MountainTrue very frustrated at the water quality in the French Broad watershed. I mean, we're a mountain stream, and we should not be having the kind of poor water quality days that we have on a regular basis. It's frustrating to me that our water quality is getting worse after so many decades of it getting better. It's frustrating to me that I can't safely take my 2-year-old swimming in the French Broad whenever I want to. I can sometimes, but a lot of times it's not safe to go swimming in the French Broad River, particularly in certain sections of the river. And so we decided we needed to do more years ago, and it wasn't okay just to continue the same old thing we've been doing to try and improve our watershed. And that's kind of how we got to where we are, doing source tracking. We just completed that infrared imaging that I mentioned, which has identified, maybe 80 or so potential sewer leaks that Anna and I will be following up on. And we're using the data now to really advocate for targeted solutions. So the solutions that we're pushing for, because of the source tracking that we've done, is increasing the budget to the soil and water conservation districts. If for those of you aren't familiar, those are the soil and water districts are the folks that provide agricultural cost share money. So if you're a farmer and you want to fence your cattle out of your stream, which is a great best management practice, you can go to soil and water districts and they might provide 70 or 80% of the match funding to do that cattle fencing. There's way more demand than there is funds right now to do that program. And so we're pushing the North Carolina General Assembly to increase those funds, help farmers do the right thing, improve our water quality. And we're really asking for folks' help too. So if you go to iloverivers.org, you'll see some of these action items. It's a way you can take action. You can email your elected officials and tell them that you think this is a priority and more needs to be done. The Clean Water Act was passed, 50 years ago, and all waters are supposed to be fishable and swimmable. And most of the time on the French Broad River, that's not the case. We're also pushing for improved infrastructure funding. So at the federal level, a key problem with our sewage is that we have aging infrastructure. In the city of Asheville, some of our pipes are over 100 years old. And so it's not that surprising when you do have leaks and you do have sewage overflows. I just looked at some of our sewage overflow numbers, and across the state of North Carolina for the last 6 or 7 months, we had over 30 million gallons of sewage overflow into our waterways. That's an unacceptably high number. And a big part of that is that our infrastructure needs improving, and there's some opportunities right now at the federal level to get local governments the money they need to do that. So, but they're not going to get there without you guys making the French Broad and this problem the squeaky wheel. So hopefully you guys can help us do that. And now's the time you get to ask whatever question you want. If it's a really smart question, then I'm gonna let Joseph answer it. If it's not, then I can answer it. I'll start. I'll break the ice. This is a question I think I know the answer to, but it was for those of you that are pondering using this method. It's a question I had for Joseph early on. We were actually comparing Jonah Ventures with some other commercial labs that do this kind of work. And, Joseph described in that first slide the way they do their DNA testing. And here's why I thought their method was better, because you're getting DNA, say, from humans. And, the first method is you're getting gut DNA. And so you're quite likely or somewhat likely to get a false negative. I mean, you go out and you take a sample and you don't find any cow, but you could just be looking for the wrong cow marker. There could be cow there, you just didn't find it. What I think in Jonah Ventures method is that you might get a false positive. You could get a hit of cow, and it might not be from cow waste. It could be from just a cow waded in a stream, and maybe some of the DNA from the cow came down. Is that fair, Joseph? I think you might be muted, or I can't hear you.
see, can you hear me now?
Yeah.
All right. Still good?
Yeah.
All right. Sorry, I was just getting error messages for some reason. Yeah, so there's, I think in general, Hartwell, the way that you explained the data is spot on. You're seeing such consistent patterns with cattle DNA across different sites and such few times that it's a significant amount of human DNA or dog DNA that cattle is probably the #1 contribution at this point. There's no reason to necessarily think about the biases or of the data. That being said, for full transparency, there are other ways that DNA can get into the water that aren't necessarily fecal contamination, right? So just as I said, if we're somebody has a garbage disposal and they're throwing scraps into that, you'll get that source of DNA without it being ever anything associated with fecal material in that sense. If somebody goes swimming in the water and some of their DNA sloughs off, you're going to get some contribution of human DNA without there actually being human fecal material. So I think we think about the biases, false positives and false negatives in that interpretation to make sure that we're thinking about all the potential competing hypotheses to explain any one particular data point, no less a pattern in general. There's other things that we can do moving forward. Some of that is spatial sampling, some of that's a technique that we haven't really tested yet. But it is possible to take a look at the DNA that we find, the human DNA, for example, and start to see. You almost do genetic approaches, just like ancestry, to see what type, who it's coming from. We can also look for more specific markers for cattle to see what breeds it is, for pigs to see if it's feral pigs or domestic pigs, things like that. So that's getting into really boutique, difficult to use, difficult to apply approaches to drill into things. And what we're doing here with the source tracking approach is really giving you a tool, just like Hartwell's used and everybody's used so far, that is relatively inexpensive, but provides data that is difficult to get through other means. And so therefore it can be applied broadly to paint a more exact picture of what's going on. That was our goal with setting up the source tracking. And so far it looks like it's been pretty helpful.
Yeah, and so my thought was I'd much rather have a false positive than a false negative, because if I got a false negative, if I got a negative, I'm probably going to quit looking. If I take a sample and it shows no cow, then I'm probably going to quit looking for cow. If I get a sample that shows cow, my little 2-year-old's in here joining the webinar. If I get cow, I'm probably going to go back to verify what level cow, was that a rain event? And so I'm probably going to figure out it was a false positive at some point. Whereas the other way I might not. Joseph, this next question might be one for you. It looks like there's one about evidence of contamination from horses. That's something we didn't look for. I don't know if that's something you can look for.
Yeah, theoretically, we can look for almost any host. We do have a horse marker that we can look for separately, and we've done that. We've done that in Florida, for example, where there's more horse farms in the areas where people have done the work. So, yeah, it's something. Quite often, once we get data like these, somebody will respond like, "How do you know it's not at another species? It's not raccoons, it's not geese, it's not deer, it's not horses." And so we occasionally will add those. But that's up to you guys to ask us to do that.
And there was another question about what advocacy methods are most effective. That's a good question. I mean, I'll just give a few examples, and Anna, feel free to chime in if you want. When we're looking for sewer and septic or agriculture, I mean, that's where most of your problems are coming for, at least in the French Broad. And, we've had pretty good luck working with our, local sewer authorities. So when we find a sewer leak, that's kind of an ideal scenario, because we'll call them and they'll come out there and they'll fix it in the same day usually. And so that's a great scenario. Septic is a little more challenging. It's definitely not a same day. I mean, there's a spot in Henderson County that I've been trying to get fixed for a year, and I think it maybe just now is fixed. So that may not be answering your question as far as advocacy. So when the systems that are set up to deal with these problems don't work, I think is kind of where we get to the advocacy issues. So, or when they're not working as well as they should. So that's kind of where we're at in the French Broad. I mean, we have systems in place. I mean, your farm can't pollute a stream to the point that it violates water quality standards, and you can't have sewage overflowing or leaking out into your septic or out of your sewer system, but it happens, and it happens all too frequently to the level that the French Broad isn't safe to swim in most of the time. And that's kind of where we're getting at with our advocacy is looking at why and what are the systems that are already in place to deal with that, and then what aren't. And so we're starting our advocacy with the systems that are already in place. I mean, there's already the soil and water conservation districts, which are, excellent, and they do a great job working with farmers. They just don't have the resources they need to do as much work as needs to be done. So if we can get them money to do more of that, that's a positive, and that's a system that's already in place. Same with, our infrastructure. There's very few sewer districts that want to have sewage flowing into our waterways. I mean, that's pretty rare nowadays. And I don't know if any in the French Broad watershed that are sort of negligent to that point. But most of them don't have the resources they need. You're looking at, can be millions of dollars to upgrade a sewer line that leaks or takes on water to the point that it overflows in a rainstorm. And so that can be hard to come by. And that's where the infrastructure funding we're looking at needs to come from. Now, going further, though, I mean, I don't think it's unreasonable to say that we think there might need to be better rules around agriculture. It doesn't seem at all unreasonable that there should be a rule that requires cattle to be fenced out of streams. They aren't currently, but we would love to go this voluntary approach and have all cattle fenced out. But if we continue to have problems in the French Broad watershed and it continues to be coming from cattle, then there may be a need to advocate for, stuff like that, like fencing cattle out of the stream as a requirement with, assistance from the state to help do that. I don't know if that answers that question, but you got anything to add, Anna?
No, I thought you hit that really well. We did have a couple more questions come in. This one might be good for Joseph. Can you tell a little bit more about how different DNA is identified in the lab? Is it through visual or a chemical process?
Yeah, that's a good question. So we have a machine is the best way to phrase it. And the process by which we look at which DNA sequences are there, gets into how people sequence DNA. But essentially in this case, we are going to focus on a relatively short region in the mitochondria of animals. And what happens is that process has two steps. The first is that it's going to have enzymes. So we'll extract DNA and then we add some enzymes that will make copies of DNA. So it doubles. And essentially every cycle that we put it through, there's twice as much DNA as was there before. And then there's also another little marker that gets stuck on there such that it will fluoresce when this DNA gets produced. And so over time, more and more fluorescence builds up. And when it reaches a point that we can detect it, so we can actually see that fluorescence, the machine can see the fluorescence, we note how long that took, and then we match that up with a standard curve that we've generated by having known amounts of DNA, and we see how long it takes for us to see that. So it's somewhat visual to a certain degree. I mean, it's chemical, like biology of life is all chemistry in that sense, but it's not a technique that's that new. These machines have been around for 20 years. We're just kind of applying it in a different manner in that sense. So that's about all I understand of it. Thanks.
Maybe a good follow-up question. Does Jonah Ventures analyze if you were to get a pure septic source Is it easy to determine if the contributions from what you're seeing is meat in the diet versus human?
Yeah, so that's uh, there's been some of this work. So we've been part of studies where half of actually what we do is we reconstruct the diets of animals when people send us poop. And there's been studies where human fecal material has been sent to us, and we've analyzed that for dietary studies. So, for example, if you're part of a clinical trial and they make you list all the things that you ate, people don't do a good job of listing that. And so they've asked us to go and sequence what's coming out of people's poop so we can reconstruct their diet. And it's going to depend. It depends on what people eat. So for example, we'd get one sample back, we'd look at it for plants and vertebrate DNA at the same time. And you get something like bell peppers. What was it? One time it was bell peppers, rice, and I think in this case it was lamb. And somebody had just had stuffed peppers the night before, right? So it just depends on what people are eating in that sense. It still would be helpful to go into septic systems and to go into wastewater treatment plants and start to get a pattern for those signatures, what the ratios are, what the standards are. We don't have a broad data set on that, so we're using first principles, but more data would always be helpful.
There's another question about basically, somebody's been kayaking in the upper French Broad and seen a lot of cows in the creek, in the river. And that isn't nearly as common as it used to be, but it is somewhat common still, particularly on smaller tributaries that run through farms. The main stem of the French Broad has that, but not as much as it used to. A lot of that is because, the work of soil and water conservation districts that have fenced out cattle or farmers that have realized that cows in our creek are a water quality impact. But it certainly still happens in the mountains of West North Carolina. It's hard because there are so many little creeks. And, if you have a farm of any size, you're going to likely have a creek cutting through it somewhere. And so fencing your cattle out of all your creeks is pretty difficult, can be difficult, can be expensive. And then even if you get the cost of that fence paid up front, you still got to maintain the fence. And that's a concern for farmers. They do, there are some good practices like hardening stream crossing. So maybe there's an area where cattle do like to cross to get to another field, and you can put in a kind of a hardened structure so they're not, tromping through the stream and they're not hanging out. And most of the time, people that study cow psychology, I don't know exactly how you know this, but folks have shown that most cows are going to the creek to get shade or to get water. So if you provide shade and water in a different location, cows are kind of lazy and they'll just take it where they can get it. And so that's another option too. You provide a watering source away from the stream with a shade area, and cows aren't necessarily going to take the hard route of getting in and out of that waterway to get. So there are different methods, and there are ways to go about it. And there's a lot more of that work that needs to be done, but a lot of work has been done. Same question around, people often ask about how many straight pipes are there still. There's still quite a few, but nowhere nearly what there used to be, thanks to the work that's been done, a lot of that by our state agencies over the last several decades.
Hey, Hartwell, we had a question come in the chat from Walter. He asks, have we been able to isolate river segments that are problematic? Do you want to give kind of a quick history of how we've done that?
Yeah, and so I should have probably hit on that a little more. Those kind of 4 spots that I showed, Cane Creek, Mud Creek, Hominy Creek, and Pearson Bridge. We picked those because those are some of our worst sites using that swim guide sampling that we're doing. So the swim guide sampling really is informing a lot of this other work. We know that those are problem sites, and so let's go sample those with the source tracking to figure out why they're problem sites, as opposed to kind of blindly picking sites and hoping that we get hits. So, because of Swim Guide, we now have, getting close to a decade in some places of data at sites in the French Broad. And so we are starting to get a sense of where some of our worst sections are. And then, like I was mentioning briefly, Hominy Creek is a good example of a site that's continued to be problematic. And because of that, we have several years of data, of upstream data, where we've kind of isolated different segments of Hominy Creek, and we know there's certain tributaries that are worse than others. And I think we found, close to a dozen problems in Hominy Creek now, ag, septic, and sewer. And so we're eliminating problems, but the overall problem is not improving dramatically. So there's more work to be done.
Thanks. That was a good synopsis. So now that looks like all the questions that have come in. Does anybody want to just give like a final thought? And it's okay if you don't.
I'm happy to try and sum it up. The French Broad River Clean Water Act was passed in 1972. And, at that point, the French Broad was said to be too thick to drink and too thin to plow. And we're now at a point where the French Broad's a thriving recreation destination with thousands of, people tubing and boating and fishing on any given weekend in the summer. But it's often not clean enough for folks to go swimming. And without, doing additional work like this, without investing the money, without improving regulations and enforcement, we're not going to ever meet the goals of the Clean Water Act. And so this is a key step in getting us there, but there's a lot more that needs to be done. And without folks demanding it, we're also never going to get there. The only thing that ever happens is stuff that people push and complain about and make the squeaky wheel. So that's why we rely on the public to help us do that work. And use science to help drive those solutions.
So Hartwell, having done a year's worth of sampling, to really nail these questions down, how many more samples do you think you would need to start to paint that picture, a three-dimensional or two-dimensional picture of what's happening in the French Broad? I mean, 50 samples, is that about right? Or are you factor 2, factor 10 off? What do you think?
Yeah, that's a good question. I feel, and Anna, you answer too. I feel at those sites where we concentrated more samples, we probably had, I don't know, maybe 6 or 7 rounds. Does that sound right, Anna? At those kind of 4 key locations, I felt pretty confident that what we were seeing there was what it is. We kind of took that broader sweep of the watershed, and a lot of those sites we only got 2 rounds of data, and I felt less confident saying, any real broad statement at some of those other sites. So I'd love to do, 3 or 4 more rounds of that watershed wide sampling, and then I'd feel comfortable in a variety of conditions. So that's probably, that's probably another 50 or so samples. Does that sound about right, Anna?
Yeah, I'd agree with all of that, especially some of the more rural areas that we haven't done a ton of E. coli specific samples on, but we know our swim guide has shown that they tend to have high levels of E. coli. It'd be nice to do some source tracking there.
And how many miles of river are we talking, roughly?
Oh man, thousands. I mean, the French Broad in North Carolina is like 117 miles, but tributary wise, I think is 2,000-3,000. So yeah, I mean, we're only scratching the surface.
We had
I would just say, to really, like, you saw what we did on Boulder Creek, which was just one, that was just a couple miles of river. Spatial and temporal resolution is really important. So kind of cranking up goals as far as the spatial patterns, I think would be interesting. Independent of, us running samples for you. I think you need to be an order of magnitude, factor 10 higher. Because really start to see the spatial pattern. I know it's hard and it's, there's a whole bunch of work that goes into it, but you can get volunteers, take the samples just as well. It'd be good to see maps.
Yeah, I would certainly agree with you. I think to get a broad sweep maybe that 50 samples, but I think, yeah, to start to really nail down what segments, and you're probably right.
Yeah, it's hundreds.
Well, thank y'all so much for being here today, and answering all these questions, showing us talking out the details. If folks are interested in what we're doing, you can follow us at MountainTrue or at FB Riverkeeper on Facebook and Instagram. You can sign on to our email list and keep up to date with all the initiatives and petitions and action alerts that we do throughout the year. But feel free to email if you'll have questions after this, and I'll be sure to send this presentation out, this recording out to folks who couldn't be here. And if you're interested, I'll just send it out to the whole list of folks that have signed up. But thanks,
Yeah,
Joseph, so much for your time.
Yep, thank you.
Yeah, sorry, Hartwell.
Thanks, everybody. Thanks, Joseph. And then everybody check out I Love Rivers, our I Love Rivers webpage. That has all of our action items based on a lot of this sampling. So
Yeah, awesome.